Today's lecture is on radiation biology. Let's start with the objectives for today. At the end of today's lecture, students will know the determinants of biological damage. Students will be able to classify radiation effects as stochastic or deterministic. Students will learn about the direct and indirect radiation interactions with tissues and the chemical basis by which radiation damage is initiated. Students will also know about response to radiation damage at the molecular and cellular levels. We shall also learn the factors affecting radiosensitivities of cells. There are multiple factors that determine a biological system's response to radiation. These factors include two main variables associated with the source of radiation and the biological system irradiated. Some radiation sources are more damaging than others, and some biological systems are more sensitive to radiation than others. To complicate matters even further, identifying biological effects also depends on the method of observation and how much time has elapsed following irradiation. Let's first learn about radiation-related factors. Radiation-related factors include absorbed dose, which refers to the quantity of radiation deposited in the biological system, the dose rate, as well as the type and quality of the radiation. Now let's look at the biological-related variables. Biological variables include tissue or cellular radiosensitivities, the type of damage, and the system's response to damage. Cellular radiosensitivity is determined by several variables, some of which are inherent to the cells themselves, while others relate to the conditions in the cells at the time of irradiation. Damage may be observed at the molecular or cellular level, but may or may not result in clinically detectable adverse events. In addition, although some responses to radiation exposure appear instantaneously or within minutes to hours, like skin reddening, others like solid tumors may take weeks, years, or even decades to appear. So once we start observing biological effects, the next task is to classify these effects. Let's look next at how we classify biological effects. There are two types of biological effects. We can first look at what are called stochastic effects. The word stochastic refers to chance or probability. So to that point, a stochastic effect is one in which the probability of the effect increases with dose, but the severity of the effect is independent of dose. Stochastic effects include radiation-induced cancer and hereditary effects. It is generally assumed that there is no dose threshold, that is, no minimum dose required to cause stochastic effects, since injury to just one cell could result in an effect. Stochastic effects from diagnostic X-rays are the principal health risk to both patients and radiology staff. The other type of radiation effect is deterministic. The severity of a deterministic effect increases with the dose delivered, but the probability of occurrence is independent of dose. As opposed to stochastic effects which have no dose threshold, for a deterministic effect to occur, the dose delivered must be above a certain threshold value. The dose threshold value is different for different effects, and also because of a wide individual variability in response to radiation, each threshold dose value is just an approximation of the dose that would likely cause a specified effect. Deterministic effects include cataracts, erythema, and blood cell effects, and acute radiation syndrome. Let's now look at radiation interactions in tissues. As discussed in the previous lecture, the interactions of X-rays and gamma-ray photons in tissue, as well as radiations emitted during radionuclide decay, result in the production of energetic electrons. These electrons deposit their kinetic energy in tissues by excitation, ionization, and radiative losses or thermal heating. Chemical changes can result from excitation and ionization, which can go on to produce biological effects. Actually, most of the energy deposited by radiation ends up as a very small amount of heat. If cell changes do produce biological effects, the damage is delayed and only becomes apparent after a time period of minutes or years. This period is referred to as a latent period. Also note that cell changes or injuries caused by radiation cannot be distinguished from cell changes or injuries caused by other physical or chemical agents. One important point to keep in mind is that observable effects such as chromosome breakage, cell death, oncogenic transformation, and acute radiation sickness all have their origin in radiation-induced chemical changes in important biomolecules. On this slide, we shall look at the ways that radiation can interact with tissues. One way that radiation can interact with tissue is called a direct effect. This occurs when a photon directly ionizes or excites a DNA molecule, as shown in the lower half of this figure. The other way radiation can interact is by indirect effects. In this case, a reactive chemical is produced by radiation interaction with water molecules. This reactive chemical species then attacks and damages DNA molecules. Because tissue is made up mostly of water, indirect interactions are more common than direct interactions. On this slide, we shall look more closely at how reactive chemical species produced by low-energy electrons cause indirect damage. Following an initial ionization event from, say, gamma rays, low-energy electrons and delta rays are set in motion. These electrons produce a unique ionization pattern over a very short range of between 4 to 12 nanometers along the path of the primary ionization track. Energy deposited along shorter tracks with fewer ionization events are called spares, and deposition on longer tracks with more ionization events are called blobs. Spares and blobs produce highly reactive chemical species called free radicals along their paths, thereby increasing the probability of molecular damage. The figure on this slide shows these free radicals and their interactions with DNA. Let's now look at the chemistry of free radical production. When radiation is absorbed by a water molecule, a positive water ion and free electron are produced. The free electron can interact with another water molecule, forming a negative water ion. Water ions are unstable and dissociate to form more ions and free radicals. The main free radicals shown on this slide are the hydroxyl radical and hydrogen radical. Free radicals are very reactive. Some free radicals will recombine to form water molecules and are therefore inactivated. But free radicals like the hydroxyls can also combine to form hydrogen peroxide. Both hydrogen peroxide and the free radicals themselves can directly interact with DNA. However, for low linear energy transfer radiations like X-rays and gamma rays, the molecular yield of hydrogen peroxide is low, and most indirect effects are due to the interactions of the hydroxyl radicals. An important point is that when oxygen is present, it stabilizes free radicals by combining with a hydrogen free radical to form a highly reactive oxygen species such as the hydroperoxyl radical. Also, the lifetime of a hydrogen free radical is lengthened if it combines with an oxygen molecule, and this enhances its potential for causing DNA damage. Once DNA damage has occurred, either through indirect or direct action, the cell may mutate and then produce a biological response, or the damaged cell may be able to repair itself. Radiation interactions and mutations or repairs can occur in microseconds, while biological response may require minutes or decades to occur. The severity of a biological response depends on what cell function was affected. Some functions are minor, while others, such as cell division, are critical. It is also common that cells with more damage die during mitosis and thereby eliminate the potential for future biological response. I mentioned a few slides back that the biological effects of radiation depends on both the type of radiation and the radiosensitivities of the cell. Experiments with cells and animals have shown that the biological effect of radiation depends also on the distribution of energy deposition at the molecular level, a type of microdosimetry. This tells us that LET is an important factor in biological response. We can evaluate the effectiveness of different types and energies of radiation and their associated LETs by comparing the biological response of each type of test radiation with the same response from a reference radiation, typically 250 keV X-rays. The ratio of a dose of 250 keV X-rays that produces a response to the dose of a test radiation that produces the same response is called the relative biological effectiveness, RBE. On this figure, we see that RBE is initially proportional to LET for X-rays and gamma rays. As the LET of the radiation increases, so does the RBE. The increase is attributed to the higher specific ionization associated with high LET radiations like alpha particles compared to low LET radiations like X-rays and gamma rays. Beyond approximately 100 keV per micrometer in tissue, the RBE decreases with increasing LET because of the overkill effect. Overkill or wasted dose refers to the deposition of radiation energy in excess of that necessary to produce the maximum biologic effect. While on this graph RBE peaks at about 8, RBE can range from 1 to more than 20. RBE also depends on the total dose and the dose rate. Let's now look at molecular response to radiation. It is thought that damage to a DNA molecule in the cell nucleus is the cause of significant radiation-induced cell damage. The current model of cell damage from radiation is called target theory. According to target theory, a cell may be inactivated by a certain number of hits, where a hit refers to a direct or indirect radiation interaction with a key molecule. Interactions with any redundant molecules are less important since loss of one of these molecules would not result in cell death. Let's look at some types of DNA damage. When radiation is deposited directly or indirectly, it induces chemical changes in a large molecule like DNA that may then undergo a variety of structural changes. These structural changes include hydrogen bond breakage, molecular degradation or breakage, and intermolecular and intramolecular cross-linking. Let's look now at single-strand breaks and double-strand breaks. These figures show part of a DNA molecule with a backbone and base pairs. One structural change that radiation can cause is a single-strand break, where one backbone of the helix breaks apart as indicated by the green arrows in the figure. This type of damage is most common for low-energy radiation. If the other strand remains intact, the break can be easily repaired and won't result in significant cell damage. A double-strand break occurs when two single-strand breaks are directly opposite on the helix. This sort of break requires higher energy radiation, such as a heavy charged particle. A double-strand break is not as likely to be repairable and may result in a DNA molecule snapping in two pieces with subsequent mutation. Other radiation effects on DNA include base deletion, where a base is removed. The hole left behind can be filled with a different base. This is called base substitution. The loss or change of a base is classified as a mutation. The DNA damages shown on the previous slides induce several cellular responses that enable the cell to repair or cope with the damage. For example, the cell may activate a checkpoint in the cell cycle. This stops the cell cycle and allows for repair of damaged or incompletely replicated chromosomes before the cycle resumes. Repair of DNA damage depends on several factors, including the stage of the cell cycle and the type and location of the damage or lesion. The figure on this slide shows the processes involved in one type of repair pathway called the short patch base excision repair. If the mutation is catastrophic, the cell may initiate a programmed cell death called apoptosis, which leads to the damaged material being eliminated. There is experimental evidence for DNA repair. On this slide, immunofluorescent techniques are used to reveal double-strand break induction and repair in human fibroblasts. Fibroblasts are shown 3 minutes after exposure to 200 milligray and also 24 hours after the initial damage. Even with double-strand breaks, DNA rejoining is virtually complete within 24 hours, as the third picture shows. When chromosome breaks occur, it is possible for the broken fragments to recombine with other broken fragments. It is also possible there is no recombination of broken fragments. In a group of irradiated chromosomes with broken fragments, recombination and subsequent replication can result in a variety of chromosome aberrations. The type of aberration depends on the configuration of the fragments. The top figure shows different breaks and the resulting chromosome aberrations that are formed. The top photograph and figure show actual chromosome aberrations caused by irradiation of human leukocytes. Row A shows a chromosome break. There is no recombination, so after replication and separation, centric and eccentric fragments are formed. Centric and eccentric fragments are shown in the bottom picture after irradiation of human leukocytes. Centrics have a centromere and eccentrics do not. Row B in the top figure shows a type of chromosome aberration caused when a single chromosome is broken in two places and the broken fragments recombine. After replication and separation, an aberration called a ring is formed. The bottom photograph shows different chromosome aberrations caused by irradiation of human leukocytes. A ring is shown. Row C shows a type of chromosome aberration called a translocation. In the first step, Two chromosomes each have one break at an end. The second step shows the broken ends interchanging, with each eccentric end recombining with a different centric fragment. After replication and separation, the two resulting chromosomes have exchanged or translocated end pieces. This type of aberration does not generally lead to cell death, but it's thought that it could lead to a normal cell becoming cancerous. The last row D, demonstrates a type of chromosome aberration called a dicentric. In the first step, two different broken chromosomes are shown before replication. The second step shows the adjacent broken fragments interchanging, with centrics recombining with centrics and eccentrics recombining with eccentrics. The third step shows the resulting chromosomes produced after replication. After separation, a chromosome with two centromeres, called a dicentric, is formed, and the two other chromosomes without centromeres or eccentric fragments are also formed. The eccentric fragments are eventually lost in the next mitosis. This type of aberration is generally lethal to the cell. There is one practical application of chromosome aberrations. It turns out the number of chromosome aberrations in human lymphocytes can be used to estimate the amount of radiation a person has received in the case of high exposures. Actually, it was common practice in the 1920s and 30s for medical personnel working with early X-ray machines and fluoroscopes to have blood tests done weekly to determine the amount of radiation exposures they had received. Total body doses greater than 250 milligray can be measured with this method. Though the number of chromosome aberrations decreases in time after an exposure, the effect persists and can be seen for many years. Let's now look at cellular response to radiation. There are a number of possible cellular responses. These are delayed cell division, apoptosis, reproductive failure, DNA mutations, and phenotypic transformations, some of which can become potentially oncogenic. A number of effects have also been observed in vitro. The first is a bystander effect. In this effect, irradiated cells or tissues can produce deleterious effects on non-radiated cells or tissues. The second effect is an adaptive response. In this effect, irradiated cells become more radioresistant to subsequent radiations. The third effect is genomic instability. In genomic instability, there is a delayed expression of radiation damage over many cell cycles after irradiation. All these responses depend on biological factors like the cell and its environment. For example, cell type, oxygen tension, cell cycle at time of irradiation, as well as on several factors related to the radiation exposure, for example, dose rate, and LET. One way of evaluating the biological effect of radiation is to measure cell proliferation following exposure and then plot a cell survival curve. This method makes reproductive failure an important effect because it can be used to study and compare different types of radiations, different environmental factors, and different types of cells. First, cells are grown in tissue culture, the cell colonies are exposed to radiation, and the surviving colonies are counted. A plot of the fraction surviving versus the radiation dose is called a cell survival curve and is a tool used to demonstrate the effect of radiation. The graph on this slide is a typical cell survival curve. Note that the horizontal axis is dose on a linear scale and the vertical axis is the surviving cell fraction on a logarithmic scale. The general shape of a cell survival curve starts with a cell fraction of 1 at 0 dose. The line curves at low doses, then forms a straight portion at high doses. Three different parameters are demonstrated on this curve: the extrapolation number n, the D0 dose, and the quasi-threshold dose dq. Let's discuss what each of these parameters represents. Let's start with the extrapolation number n. It is found by extrapolating the linear high-dose portion of the curve back to a dose of 0. It is a measure of the shoulder of the curve. The larger the n, the wider the shoulder of the cell survival curve. Generally, the extrapolation number for mammalian cells falls between 2 and 10. The theory is that this value represents the number of hits required to inactivate a cell or the number of critical targets within a certain type of cell. The D0 dose describes the radiosensitivity of the cell population under study. The value of the D0 dose is the reciprocal of the slope of the linear portion of the cell survival curve. If a cell is very sensitive to radiation, then the slope of the survival curve will be steep and the value of D0 will be small. If a cell is radioresistant, the slope will be rather flat and D0 will be large. The D0 for mammalian cells is approximately 1 to 2 gray for low LET radiation. The value of quasi-threshold dose dq is the width of the shoulder region of the curve. It is determined by extrapolating the linear portion of the curve back to zero dose and finding the dose value corresponding to a survival fraction of 1. The quasi-threshold dose is a measure of the ability of cells to recover from sublethal damage. A large dq value indicates cells can easily recover from radiation exposure or that the exposure is low energy. Here is a plot of survival curves for several cell types. Notice the wide variability in response to radiation exposure. By looking at these curves, we can tell that mammalian cells, curve A, are more sensitive to radiation than, say, yeast cells, curve D. Cells with the highest quasi-threshold dose have increased ability to repair damage from radiation exposure. So one may ask why there is such variability in the response of different cells to radiation exposure. There are a number of different factors that affect the way cells respond to radiation exposure. Some of these factors are related to the radiation itself, such as radiation dose rate, dose fractionation, and radiation quality. Other factors relate to the environment surrounding the cells. The presence of oxygen increases cell damage, and the presence of various chemical protectors can decrease cell damage. In addition, different types of cells respond differently to radiation, and cells are more sensitive to radiation damage during different parts of the reproductive cycle. We can look at each of these factors in more detail in the next few slides. Let's look first at radiation dose and dose fractionation. For the same amount of radiation, more biological damage is produced by radiation delivered at high dose rates compared to low dose rates. Similarly, if the radiation dose is separated into different sessions with an interval of days or weeks, the amount of damage is reduced for the same total dose. The top figure shows the dose rate effect, and the bottom figure shows the dose fractionation effect. Notice the broader shoulder of the low dose rate radiation, indicating its reduced effectiveness compared to the high dose rate radiation. The mechanism for this effect is that when radiation is delivered slowly or in fractionated sessions, cells have more time to repair sublethal damage such as single-strand DNA breaks. As these figures show, with more time for repair, the shoulder of the cell survival curve increases, that is, the quasi-threshold dose increases, and the slope of the linear portion becomes shallower, that is, the D0 dose becomes higher. This technique is fundamental to the practice of radiation therapy. The intervals between doses may be hours to a few days, and this allows the repair mechanism in healthy tissues to gain an advantage over tumor by repairing some of the sublethal damage. The next factor is radiation quality. Radiation quality is described by the linear energy transfer, or LET. LET is defined as the amount of energy deposited per unit distance for a particle or photon traveling through matter. High LET radiations include protons, high-energy photons, and larger charged particles such as alpha particles. Low LET radiations include electrons, beta particles, and low-energy X-rays and gamma rays. Relative to biological damage and for the same dose rate, high LET radiations will cause greater damage to cells than low LET radiations. This is because high LET radiations are more likely to cause double-strand breaks which are not easily repaired. This figure shows how as the LET is increased, the curve becomes steeper. And you can see the steepest curve is for alpha particles while the shallowest curve is for X-rays. Another factor affecting radiosensitivity of cells is the presence of oxygen. If free oxygen is present during radiation exposure, it can increase cell damage. As we discussed earlier for indirect interactions, free oxygen molecules can combine with the resulting free radicals. Once combined with oxygen, the free radicals are stabilized and cannot recombine to form water and neutralize. With increased number of free radicals comes an increase in DNA damage. The relative effectiveness of radiation to produce damage at various oxygen tensions is described by the oxygen enhancement ratio. Oxygen enhancement ratio is the dose at hypoxia divided by dose in the presence of oxygen for an effect. The graph shows the oxygen enhancement ratio for low LET and high LET radiations. For low LET radiations, if you select a survival fraction of 0.1, you see that the dose at hypoxia is 6 gray and the dose in the presence of oxygen is 2 gray. So the oxygen enhancement ratio is 6 divided by 2, which is 3. Research has also found that some chemicals act as protectors against radiation and can decrease cell damage. Another factor affecting cell radiosensitivity is the type of cell itself. There is a law by two French scientists that captures the fundamental characteristic of cells that affects their relative radiosensitivities. The law of Bergoni and Tribondeau from 1906 states that radiosensitivity is greatest for those cells that have a high mitotic rate, have a long mitotic future, and are undifferentiated. With only a few exceptions, for example lymphocytes, this law provides a reasonable characterization of the relative radiosensitivity of cells in vitro and in vivo. The list on this slide includes examples of cells ranked according to their sensitivity to radiation. Cells such as spermatogonia are rapidly dividing, so are quite radiosensitive. Muscle and bone cells do not divide and are highly differentiated, making them more radioresistant or least sensitive to radiation. In 1968, Rubin and Casaret refined the classification and defined 5 cell types according to characteristics that affect their radiosensitivity. These cell types are shown and the meanings are below the table. Again, one notable exception to these general rules are lymphocytes. By the classification, they should be radioresistant since they never divide, but actually lymphocytes are one of the most radiosensitive cells in the body. The last factor affecting cell radiosensitivity on our list is the phase of the cell cycle. Cells are most sensitive to radiation damage just before mitosis begins and during mitosis. The least radiosensitive or most radioresistant portion of the cell cycle is the late S phase when DNA is synthesized. Cells are less sensitive during the preparation period for DNA synthesis or G1 phase. Let's end with a few questions. First question, which part of the cell cycle is least sensitive to radiation damage? Your choices are late S phase, M phase, G2 phase, G1 phase. The correct choice is late S phase. Next question. The fundamental characteristics of cells that affect their relative radiosensitivities was discovered by Bergoni and Tribondeau in 1906. Which of these is not a characteristic? Your choices are rapid divisions, large number of future divisions, low differentiation, adaptive response. The correct choice is adaptive response. Next question. Select the correct choice to fill in the blank. In the presence of low or high LET radiation, free oxygen blanks cell damage. Your choices are first choice, has no effect on. Second choice, can decrease. Third choice, can increase. The correct choice is can increase. Next question. The number of chromosome aberrations in human lymphocytes can be used to estimate the amount of radiation a person has received in the case of high exposures. Is this true or false? The correct choice is true. Next question. Which part of the cell cycle is most sensitive to radiation damage? Your choices are late S phase, mitosis, G2 phase, G1 phase. The correct choice is mitosis. Thank you for watching this presentation.